Hey, hello, this is Elim Romero, and I promised to bring a video to install the scatoolbox from Dr. Sai's lab. So the steps are like this. First, we open MATLAB. I have it open already because it takes time to load. So right now it's just open. And right after opening, the first way to download it or installing it, it's by clicking in the Adds On. And this is gonna bring the official MATLAB installations. So we look only for scgea toolbox like that, and it's gonna be that one appearing. That stands for okay, remember my selection, and okay. Then this GEA stands for gene expression analysis. So we can click on add and it's gonna install it. And that's gonna be it. It's gonna take time, but I'm gonna show you the second way to install it, and that's gonna be like this. You type in scge toolbox in Google browser and it's gonna appear these first three. The first one is gonna be the official MATLAB website, which tells you recommended adds on here. We click on it and it says, oh, just look for the adds on and install it. And after installing, type in this scge toolbox to launch it. But the installation I'm gonna do it's for GitHub way. So we're gonna do this, but from the documentation. So if we go back here, we have the official GitHub. That's not gonna be the one you use, but here is the official repository that you can download from here. Likewise, it tells the steps here. So all good. We can keep it in mind. And then we got to the official website. Then here's the documentation. This I recommend because here are the cook and recipes. So the first thing is quick installation for developers. Then we go for this and we copy all these guys here. And we got to the command window and paste it and hit enter. And it should download it here in the MATLAB location. It's gonna take some time, so I'm gonna stop the video. All right, after it finishes, it's gonna say nothing, but it's gonna be finished. So you type scge toolbox and should pop out. Might take some time, it's preloading. And then we have the toolbox install, as easy as that. So we do import, and we're gonna try the load example, and we're gonna use the row just for the first test to see that everything is functionally, and we're gonna look for the 1, 2, 3 button. So that should be into here. Okay, I couldn't find the 1, 2, 3, 4, 5 button. So we are gonna do it one by one. The first one is gonna be this third in the button, and it says filter genes and cells. So we click on it and we do the basic QC. Okay, and we're gonna do usually stringent, depending if we lose too much genes and cells. Most of the time we wanna keep most of the genes, so we do relaxed in that case. But if we wanna have the cleanest, just stringent. And it's gonna be the setup, the automatic. We just click OK, and it's gonna do it. No cells and genes are removed from here because it's already cleaned up. The next thing we would like to verify is here in these label cell groups, the fourth button in the button. So we click on it and we're gonna have these classifications. One of the first to check up is batch ID. We don't want to confound our different samples. So we click on it and we see two samples here, the 14 weeks and the 08 weeks. Usually we need to import and name those ourselves, so we gotta be careful with that. And here we have other like cell type, but there is no cell type associated still. And cluster ID. Cluster ID, nothing is in there. The next step we gotta do is to do UMAP or TSNE here in this button. no, this one. Embedding cells using TSNE, UMAP, fade, whatever. So we click on it. We're gonna do UMAP 2D and hit OK. And yes, here it depends what you wanna look. We might want to explore both of them. 2000 highly variable genes. I'm gonna explain what those are later and what all genes are. In this case, I'm gonna use all genes because it's a small data set with few cells and few genes. So I'm gonna do all genes. And this is how it looks. And if we go back here into the label, we hit into batch ID, we're gonna see the two batches in different locations. And wait a second. All right, I found it. The next button is to find the clusters. The clusters are gonna try to classify the cells by vicinities in algorithms. Those I'm gonna not gonna talk in depth, but gonna have a grasp later. But what those are doing here is this button, clustering using cell embedding. So this is the k-means and relies on the UMAP representation. For now, we're gonna use this, but I have another algorithm that we are going to use later. So far, we're gonna do k-means here, and 20 clusters, that's the recommended, we just do it. And that's it, those are the classifications. In this case, if we click on the label and show cluster ID, we're gonna have those numbers are not visible, came out. So we have all the different clusters, and potentially those are different cells or different cell states. All right, the next, beside the clustering, we have the cell type assign. So we are gonna assign the cell types utilizing a curated data set from Dr. Sai's lab. And here is gonna utilize the curated by PangLaoDB. PangLaoDB is a really nice web page I'm gonna walk through later, but so far we're gonna just use this. So we hit on this and we utilize for a given sample. I think this is mice, so we're gonna use mouse and yes, automatically. And then it's gonna do the process of trying to look at those clusters and assign a cell type according to the genes in each cluster. So far so good. We merged them in this case. In some other cases, we are not gonna do that, but so far so good. This is what we got. It looks good. So we have really good separation within cells in this case and within colors as well. And that's it. The final and last step is gonna be after annotation. We're gonna go here, and this is the save button. And then we're gonna save it in SCE math file and save it here. This is the name default. You can change it, but for now, just gonna save it like that. And then after save, I can close this body and I'm gonna clear all the MATLAB memory. But if I look for it and double click, right, sorry for that. So I found that if you click on here, it's gonna appear and it's gonna be in my current working directory. So you double click on it and it's gonna say something about the loading, and I can type in scgeatoolbox parentheses SCE. That's the usual way to open it. The other way is scgeatoolbox. You click on it and then import data. Then I want to look for a math file. Hey, where is that? Over here. Click on it and should have all my process already. Cell types. Great. We look for cluster IDs. Great. So far, so good. This is how we process the samples, and later on we're gonna work in different ways to further refine this process. There is only one last mention here. I want to mention that here is a beautiful button for initial processing. Here is a running guide, single click cell type annotation. If we click on it, it's gonna do from, I am not so sure if from cleaning, QC filtering, but it's gonna do the annotation, UMAP, and everything So you can tell it, oh, which one's representation do you want? I want estimate cell cycle, UMAP, TSNE, and differentiation potency. Why not? Okay, and it's gonna work on it. I'm gonna stop the video because it might take some time. So far, so good. It finished. The thing here is that now it has way more information than probably what I would like to have. So I just click in the UMAP, yes. Now I have TSNE 3D, UMAP 3D, but I don't have the UMAP 2D. It's just fine. We can look at this and we have it again. And that's it for today, and that's how we can process the samples and look at the cell type classifications later. Thank you for just viewing the video. I hope you enjoy it, and we'll see you in next videos.